And by spatial temporal regulation, what I mean is having this temporal resolution to understand how a protein is moving around doing what it is supposed to do. How is that protein acting as a function of whatever perturbation you're throwing at it? When is it doing that? It's like trying to build a Google Maps with a lot of resolution at the cellular level.
Hi, Amine.
Hi, Tarek. How are you?
Good, and you? How are you doing?
Very good, thank you. Thanks for
California.
having me. Well, it's a bit early in the morning here, so it doesn't look like California yet. But yeah, it's great.
It looks like Boston.
Yeah.
Okay, I mean, so let's start with the papers.
Sure.
Right. So before we start with the papers, your specialty is microscopy.
Yeah.
Yes, and you've been using microscopy for many years now, applying it to different topics. So if you could very quickly give us just an overview of the different topics you use microscopy for.
Yeah. So I would say, my specialty is microscopy, but I think the focus has been on method development to try to bring together microscopy techniques or even develop microscopy techniques that allows us to understand the spatial temporal regulation of whatever protein we're interested in. And by spatial temporal regulation, what I mean is accessing a level of spatial resolution that allows you to map out protein spatial distribution, having this temporal resolution to understand how a protein is moving around doing what it is supposed to do. Even understanding self-association using Forster resonance energy transfer-based method, FRET, and we'll be talking about that. So, yeah, there's been quite a bit of variety, but I think the common denominator has been less break through the diffraction limit and less understand at a more granular scale how whichever protein that we're interested in the moment is actually doing what it's doing. So it's been an exercise of where is that protein, how is that protein acting as a function of whatever perturbation you're throwing at it, and when is it doing that? So it's just getting a bit more context. It's like trying to build a Google Maps with a lot of resolution at the cellular level.
Inside the cell.
Inside the cell.
Yeah, that's great.
And we're so far away from doing that, but we're trying.
So what do you mean by the diffraction limit?
Yeah. So the diffraction limit is this it's been defined by this guy called Abe, and it basically it has to do with the fact that intrinsically light is a wave and a particle, and as it interacts with the system that you're working with your optical components and whatnot, it will yield whichever small emitter you're actually interested in capturing. Will end up being resolved with a precision that has to do with the numerical aperture of your objective, with the excitation light that you're using to actually look at it. And at the end of the day, what you're stuck with is that you in your detector, in your camera, you end up having, even for something very small, a disk that is about half the size of the wavelength that you're using to excite your sample. So what it means is that if you look at the green fluorescent protein, despite the fact that that is very tiny, and now you look at a few GFPs that are very close to one another, instead of seeing discrete dots, what you end up seeing is a bunch of blurs that are merging next to one another, which is in many ways good enough as a way to say, well, there's more expression, less expression. These things are clustered. These things are forming, I don't know, whatever large structure in this cell type But when it comes to be able to understand how many are there, are they forming any sort of small cluster, then you can't really resolve that. And, the diffraction limit exists. You have to circumvent it. You have to go around it. It's not as if you can change the natural properties of light. And a lot of work has started, I would say, since the early 90s to try to do that. And it has all crystallized around 2014. 2014 was when a Nobel Prize was given in chemistry to Eric Betzig, Stephen Hale, and William Morner for collectively finding solutions to address this diffraction limit. And there is a set of technique that I've been working on for a while that has to do with single molecule localization microscopy. So, going back to that GFP thing I was talking about, where you cannot resolve individual ones, the trick is, what if you were to have a photoactivatable GFP? Something that you can switch on and off randomly, stochastically, during your acquisition period. And then what you end up with is discrete point spread functions. Those are the blurry disks that you're actually getting. And then, as you look at those disks, and if you draw a line profile over it going through the center, what you end up with is something that vaguely looks like a Gaussian. So then you fit an idealized 2D Gaussian to this as a way to identify the centroid, so that you can get a coordinate. And you identify that centroid with a precision that is proportional to your background and to the number of photons that you're collecting. So the higher the number of photons, the higher the spatial resolution. And a lot of this circumventing the diffraction limit has to do with improvement, as I alluded to, with the photoactivatable JP, improvements in the palette of available fluorescent proteins and organic dyes, in addition to cameras. So the EMCCD camera, the electron coupled charge device, being able to really improve the signal, or the CMOS cameras as well, that have really made the work of being able to detect really faint signals, so that you can actually do the fitting and whatnot that I was talking about.
Okay, so when you say centroid, do you just how do you calculate it? Just by doing the average?
Say that again.
When you talk about the centroid, like you fit that Gaussian, and then how do you calculate it? Just by calculating the average of all the different points? Like when you fit your Gaussian, then you have your distribution, and then just like the theoretical average of the distribution.
Yeah. So I think there is initially, I think, an exercise of detection. So what in your image, you're going to end up having multiple point spread functions. So I think the first thing is what constitutes a point spread function. Being able to understand what is the intensity threshold within which you want to operate, knowing that your point spread functions are isolated enough from each other. There are methods by which you can try to do the fitting exercise despite the fact that they might be slightly overlapping or in close proximity. Then I think, I don't remember the exact specifics on the algorithms, but through the use of linear least square or maximum likelihood estimation, you can eventually get to the point where you do that fitting in a way that is statistically coherent.
I see. So it's probably some expectation maximization.
Yep.
Okay, good.
But you'd have to talk to my version from 10 years ago to tell you the specifics.
It does make a lot of sense. Probably we should tell people what GFP is.
The green fluorescent protein. Yeah.
Yes. GFP, green fluorescent protein. Exactly.
Yeah, no. So thanks for asking that question, because I think you're going to open a tangent right now. But what I love about GFP, which, I think when we say GFP, in a way, we're using it as a placeholder word to be talking about a variety of different green fluorescent proteins. But yeah, it's been characterized and identified by Roger Chen, the late Roger Chen. And it was linked to this exercise where people noticed that jellyfish was actually emitting light. And people were like, well, what is going on here? And I think it started from this very fundamental exercise of trying to understand this natural phenomenon of a fluorescence in a live being. And eventually, I think the tour de force was, first of all, people were allowed to do that investigation because on the surface, it doesn't sound like grant money should necessarily go there. And I think that's the beauty of it. It's like, I think in my mind, the GFPs, the Taq polymerases examples are great examples because those are undertakings that allowed people to go and explore, not knowing what they would get in return. Mm-hmm. But the beauty of it is, yes, nature does what it does, and then we realize that there is this beta barrel, that is the structure of a GFP, is actually able to fold on its own without any chaperones and whatnot. And what that means is that when you express it in whatever system you're looking at, being a bacterium, a eukaryotic cell, well, you're actually going to get the inherent properties of that thing. And, it's a beta barrel with a chromophore in the center. And with GFP, you typically excite it with a 488 excitation. You get an emission in the 520 or something like that. And that allows you now to, through genetic engineering, to place it on top of whatever protein you're interested to visualize and localize. And as you know, GFP was the starting point, and then people have done a lot of optimization and mutational optimization to look for proteins in different colors, proteins that do fold faster, that have a better maturation time, proteins that are more photostable. And now we have a really large palette. So I think, while GFP was essential, what is a fraction of labs that are still using the original GFP? Very small. People now use eGFP, the enhanced GFP, or there's a cornucopia of fluorescent proteins that have appeared that just completely blow it out of the water.
Yes, they're all different wavelengths, all different colors. Yeah. Yes.
Yeah.
I love the story of GFP because it started out as a fun fact.
Yeah.
Not as something that would completely revolutionize the scientific techniques and instruments, but it's exactly what it was.
Yeah, and I think it's a strong reminder that you have to allow people to explore, even though you might not see the return in investment.
Yes.
Tomorrow.
Exactly.
Yeah.
Exactly. I mean, modern biology would be possible without GFP.
Yeah.
I think we can probably say that. Yeah.
Yeah.
And when you say like stochastic GFP that you can turn on and off, do you have an idea how that works?
Yeah. So when it comes to let's talk about this method called photoactivation localization microscopy, and that was pioneered by Eric Betzig, who's now here at UC Berkeley. So what you do is that you're using, you're basically first photobleaching your fluorescent protein, and then you're activating it with very short pulses of violet light, 4 or 5 nanometer excitation, and very short and also very low intensity. And I don't know if you remember these Jablonski diagrams that describe the energy state. But basically what you're doing is, so you're basically getting your fluorescent protein into a long-lived off state, and then you're igniting it for a moment into a short-lived on state, and through very good hardware control, you're getting that dance of long off and short on to be one that allows you to have sparse emitters. There's also, in the case of not fluorescent proteins but organic fluorophores, there's this thing where instead of being in a normal part of the Jablonski diagram with like the E0, E1, E2 of the energy state, you're actually taking your fluorophore into this I forget what it's called, but like this triplet state. And there it's like really blinking rather than actually going from being photo bleached to being activated. It's slightly different.
Okay.
Yeah.
If I'm simplifying, what you're looking for is basically flashes.
Yeah.
Yeah.
And you're modulating that with the astute use of your lasers. And in some cases, and we'll see that in some of the papers, you're basically creating an environment within the cell that allows you to it's a lot of chemistry to actually get that off state to be predominant over the on state.
Yes. Is it because you so basically if I'm trying to understand it because I'm not a microscopy guy. So you create basically a system that you have to excite. You give it a certain amount of energy before you get that burst of light.
Yeah.
Okay. And using chemistry, you're basically modulating how much excitation it needs.
Yep.
Okay. So it's basically like resistance to light excitation through a laser.
Yeah, you could say that. I think it's a good analogy.
Okay.
No, it's good. No. And myself, while my thesis was in many ways about that, I think the fundamentals are really deep and complex, and I think that there are a lot of subtleties there.
Yeah, I can imagine. I can imagine. Do you want to go very
myself,
quickly?
I don't remember fully in depth, to be honest, but yeah.
But do you want to go very quickly about where you thesis? Where did you do your thesis?
Yeah.
That was, yeah.
So that was in 2013. I moved to Toronto, to the University of Toronto, where I worked with Christopher Yip, who's now the Dean of Engineering at the University of Toronto. So the focus of the lab was really on tool development. So, I had just come from an undergrad in biochemistry from King's College in England. And I walk into this lab and I'm told, well, why don't you go build a microscope? Pretty much. And I'm like, okay. And so that was University of Toronto. And the idea was to build one of the first, this was a combinatorial super resolution microscope. And by combinatorial, the idea was to have confocal, FRET, super resolution, and even atomic force microscopy, which I didn't do and didn't use. But and the focus, really, the biology came through collaborations. It's not as if we had a centerpiece of biology in the lab. So one of the papers that we'll discuss, that was through very close collaboration with the lab in molecular genetics. And we often use their biology as a model system, as a way to build our methods. But at the same time, on the other hand, you had labs that came and worked with us with very defined and advanced biological questions. So we're not anymore just using that as a model system, but like coming with a bottleneck of, if only I could see better and have the temporal resolution, I would understand how this thing gets shuttled from point A to point B and who are the players and whatnot. So there was a lot of that in that lab. One thing that I didn't realize the value of until more recently during that PhD experience is that we spent a lot of time troubleshooting all sorts of things. So, we would get lasers that at times were maybe deficient. You have to go and figure it out, maybe turn yourself into an electrical engineer for a couple of hours. There was just a lot of reacting and trying to make the system work. And that was very useful for me later. So that was overall the experience.
Building the machine. I'm also very big on that.
Oh yeah.
The technicalities. I'm obviously I'm not doing microscopy. I'm doing AI, but the core technical aspect is still computer science. And I really believe that students should, at the PhD level, should be very comfortable with the computer science aspect, but also the system administration and all that. Because if you can't troubleshoot the machine, you're basically out of work, and you can't always rely on other people to do that kind of stuff. So I'm really happy that you're mentioning that, because this is something that tends to be overlooked. In science, we tend to talk a lot about the results, but we don't necessarily talk about what it takes and the technical knowledge that it takes to actually get these good results.
Yeah, I totally agree, and I would even go further to add that method development is arduous and often, I think, not as celebrated as it could be, because oftentimes the applications that make those discoveries is what ends up being seen as most valuable. The enablement, that where you have to go through a crazy, tortuous journey, that once it's there, it's there. I think it partially gets taken for granted.
Yes, I agree. And the longer it's there, and the more it's taken for granted.
Yeah.
Yes, but we absolutely need methods development, because methods is actually what allows us to get the resolution to measure, which is what allows us to make the discoveries at the end.
Yeah. And I think just having a grad school experience where you're not necessarily just racing to be generating the next most important biological result, but one that's far from being linear, one that allows you to just try things and to some degree dabble in a lot of fields. I think that's a strong training, and it's going to put you in a better position later on.
Yes, I think so. And I think for that reason, I think the optimal number of years for a PhD would be at least 5, in my opinion. Because you need time to explore, you need time to try different things, you need time to mature as a researcher, and that covers so many different aspects beyond generating results.
And I totally agree with you. I mean, if I think of my first 2 years of my PhD, what was I doing? I mean, you don't know to what degree you don't know what you're doing during that period of time.
Oh yeah, that's true.
And that journey through the desert, where you have a few mirages that you think are valuable, is so valuable. I think it has the potential to shape you in a good way.
Yes, I totally agree. I totally agree. That happens several times during your PhD, which is also interesting. It's like the levels, like the illusion becomes harder. They also become better and better. So you have to deal with stronger illusions, and I think this is extremely valuable. Extremely valuable as a growing opportunity.
Yeah, I think you have to build the resilience, the realism, to just approach your work in a way where you're not. Sadly, you're driven by it, but you don't want to be as emotionally involved, so that it allows you to just approach it. You're never objective, but somewhat objectively.
Somewhat objectively, yes, and also learning when to let go.
Yep.
I think that's the hardest part. Like learning when to let go of some ideas, of some potential results, because it's not a good idea yet. Like realizing that not all your ideas are good, and being able to assess which ones are worth following. I think that definitely, in my opinion, for most people, it does require more than 3 years.
Yeah, I agree with you, Tarek. I think for me, I would say personally, the 3rd and the 4th year were like the part where I could feel a lot of growth. I could feel like the fruits of the long desert walks were starting to bear. And probably the 5th year is when you start being in control.
Yes.
But that's the time to wrap up. But I agree with you. I think, but we should make sure that it doesn't also turn into a 7-8 year long PhD. I've seen that with a few people, and that's not a beautiful story.
It's not. I mean, mine is a bit different. Mine was 7 years.
Oh, sorry.
Yeah, no, it's because mine is a different story because I started off as a computer scientist.
Mm-hmm.
And I went to work for my PhD. I was surrounded by biologists. I was in a biology lab, in a hematology lab, and I didn't know anything about the biology. So I had to learn all that. So that explains the 7 years. I still remember like my first lab meeting, all the students were presenting. Back then I spent the whole lab meeting on Wikipedia, like, looking at every term. So it did take me longer than expected, but that's an artifact of the fact that I completely changed domains for PhD. I went from hardcore computer science, anyway, applied to music, to applied to biology, and I have to say that it is great, but I have to say that I underestimated the complexity of biology.
I think we all do. I think we haven't even started understanding how complex it is.
I think so. But
we're good at telling stories as biologists.
Yes. Yeah.
I mean, and that's a problem at times. We can talk about it if you want.
Yeah, we can talk about that later. But I think it is a fundamental mechanism of how biology evolves. It's these stories is basically what allows you. They're not always true, but they're true enough to allow you to move forward. That's
true.
Yeah. And that's I think it's the beauty of biology, and that's why I keep doing it, is because you have this. It's actually it's way more abstract than even biologists realize.
Yeah.
Yes, it's a very abstract science when you think about it, because most of the thing we're talking about, we barely have the resolution to measure them. So we have to fill in the gaps with basically imagination and
words
and words and like finding the best theories that would explain the phenomena that we observe. And so obviously there's a limit to every model, because no model is true, but that allows us to move forward until that model breaks, and then we find a new one and a better one, and we keep going.
Yeah.
So it's
that's true.
It's a fascinating science, and I can say that as a computer scientist, I think as someone who started as a computer scientist, that aspect is usually severely overlooked by computer science. When people move from computer science to biology, there's always this mirage of, I have something that I can apply to images or like NLP, so it's all data is the same, so I can just apply it to biology. That never works. Biology is way too complex.
Yeah.
Way complex.
You see that a lot right now in the Bay Area, with every company wanting to slather some AI on top of some poorly defined biological hypothesis, and you're like, wait a second. It's not as if we've all been dabbling in this, and now you're going to unclog it by finding by making a better virtual model. I think the key is that you need to still capture tons of measurements, orthogonal measurements, using as many techniques as possible, curate the hell out of that, annotate it, so that eventually you end up with something that is a better model, as you said, but still will fall short in many ways at capturing the whole complexity and glory of a biological system.
I think so. And like if you take the analogy of a book, you want to do NLP on natural language, you have a book, the book is complete. I mean, you have the book, you can read it. Every letter is there, every space is there. Biology, you barely have the concept of the book. And this is why you have to take, as you said, orthogonal measurements that usually we call modalities, but you measure it from different ways. You do different statistical tests. So you just try to get a picture of what actually it is. And for most of systems, we're very, very far from having for complete systems. Obviously, if you're taking just two things and you isolate them, you can measure what's happening. But for a complete system like a cell, we're very, very far from having a precise image of what's actually happening.
Completely. I mean, with in vitro measurements, you can get a good sense of how two proteins are interacting. We've crystallized proteins. We've deposited the structures on PDB. AlphaFold has been able to capitalize on that and be able to predict to varying levels of confidence structures of things for which we don't have structure. But yeah, I think the complexity happens once you look inside a cell, and when you try to look inside a living cell, not a fixed cell. Yeah.
Yes, and we're not even talking about cell-cell interactions.
And we're not even talking, and then you take that to tissue level, organ level, person level, and I think that's why it's so damn hard to make a drug.
Yes, yeah, it's super hard. And all the different ways, it's like the different ways cells communicate. They can send electrical signals, they can send hormones, they can send exosomes. There's so many different ways. They can just touch and the receptors interact. There's so many different ways they interact. And we're barely scratching the surface of all the potential interactions between cells.
Yeah.
Yeah. So did you want to move to papers?
Sure, let's do it.
Should we start with the two-dimensional Slater swimming of sperm with a micrometer of the surface?
Yeah, let's do it.
Okay, let's start with this one.
Hey everyone, this is not an ad. Go to blueword.com/in, that's BLUWR.com/in, like inside, and write about something that makes you think. Can you see me?
Yep.
Perfect.
I mean, I can't see much without my glasses, but I can see what you're showing me.
I can zoom in if you need to. If you need me to zoom in, just let me know.
It's all good.
Okay. So this one was published in 2015 in Nature Communications. Yeah.
So that was my first paper. And as a new grad student, I had the opportunity to work closely with the first author, Reza Nosrati, who is now he's running his own group at Monash University in Australia. And in this opportunity, what Chris, my PhD advisor, told me was like, just make sure that this guy is able to run some very clean TIRF microscopy experiments. And TIRF stands for Total Internal Reflectance Fluorescence. And what you're doing essentially is that you're getting your laser, your beam, your incident light to come in through your objective at an angle known as the critical angle, for it to bounce in a manner that's only going to allow, it's not going to be shining directly at your sample. It's going to create what's known as an evanescent wave. It's very, very thin wave in theory, if you characterize it the right way. And it decays exponentially, so that if you implement it right, you're only exciting about, I'm going to use a hand wavy word, I don't know what we put in the paper, but like, let's say 200, 300 nanometers. So it's exceptional for looking at, say, at plasma membrane proteins. And people have done a lot of work using total internal reflectance fluorescence to study the spatial distribution of membrane proteins and rejecting signal from the cytoplasm and the nucleus. And of course, the devil being in the details. By no means are you capturing the membrane alone, which is much thinner than that, and to do your characterization, you want to look at things like early endosomes, for example, and you want to, since they are going to be close to the membrane, and see to what degree your well-calibrated microscope is actually capturing them or not. And, so here, what we did with Reza was Reza is a mechanical engineer looking at this. In a lab that I think there's a lot of microfluidics, is interested in partially in questions related to in vitro fertilization. So what they were interested in is how do sperm cells move, and do they actually move differently when they're closer to a surface in a viscous environment. And the relevance of that is because sperm cells have to go through quite a journey to make it to the egg. They have to go through the fallopian tube, I think, if I remember correctly, which is a pretty constricted viscous environment. And the fundamental questions that they had is, if we were to drop the sample in a dish and put it on top of a microscope, and we were to look at the swimmers that they call them, I think in the paper, bulk swimmers, versus those that are closest to the glass, do we see distinct ways of locomotion? And what this paper demonstrated by implementing that TIRF imaging is that the bulk swimmers were showing this corkscrew motion where the head was rotating while the tail was also rotating in a different way. And that's how they were propelling themselves. Well, as you're closer to the glass, as the title indicates, you get this slither swimming, where you no longer have that rotation. You just have this 2D motion of the head and the tail, which actually allows it to propel itself faster for short moments of time. So that's really what we were able to demonstrate in this study. And I think, I'm still in touch with Reza. This was an important paper that in many ways, was a fundamental building block to a lot of the papers and research projects that he's put in place now at Monash.
Okay. So just to make sure I understood correctly, so what you're saying is when closer to the glass, which means like farther from the surface, they move more like a snake.
Closer to the glass, meaning you're closer to the objective, you're closer to the surface.
Closer to the surface. Okay.
Yeah.
Okay.
Yeah.
And
it's a snake-like motion. That's right.
It's a snake-like motion, and farther from the glass, it moves more like a torpedo.
Exactly.
Okay.
And there is more subtlety to it. I think if you go through the paper, you'll see that. I think we looked at bull sperm and human sperm, and you see that in one case, one of them is actually inclined to be turning clockwise, while the other one is inclined to turn anti-clockwise. And I think that has probably provided Reza with a lot of open questions that he has or is investigating. But the goal of this for him was, with this information, can you now rationally select for performance sperm cells as a way to just improve the outcomes of IVF?
I see.
In my case, what it meant is when Reza showed up with samples, I had to make sure that the microscope operated the same way each and every day. So it was an exercise. So it was great as an introductory exercise during my PhD, because it was simply make sure that the lasers are what they're supposed to be, measure the power every day, make sure that that turf angle that I was talking about is at the turf angle that we had last week. So to me, it was really an exercise of reproducibility and just providing the collaborator with a platform that they can trust.
Which is number one most important thing in science, being able to reproduce your findings.
Yeah.
Yes. So which figure do you suggest we go to now?
I think there's one that just summarizes it in a high level.
Yeah.
I think with this one, that's fine actually.
Okay.
I think this one shows you the concept of total internal reflectance fluorescence, and it shows you. Let's see. Yeah, so I think you have your sample. You have your objective with oil immersion. So to couple that to the refractive index of your sample, and what it's showing is that. You have that critical angle, and really what you're doing is that you can select for the swimmers that are close to the glass or those that are further away by changing the angle of your incident beam. And you can if you want to select the bulk swimmers, you just have to what we had to do was to move a mirror slightly, just translate it. And before you know it, now you're shining directly through the sample. You're capturing all of them. Or you could have it in an oblique fashion, something known as high-low, where you'd be capturing less of the ones at the very top of the solution. So in this paper, we just did a few. I think if I remember correctly, we measured in turf and not in turf to be able to select.
Wow. Okay, that's.
But that's all there is to it, without wanting to take away from the beauty and complexity of what Reza did.
Nice. And in figure 2, I think this is where you measure
That's right.
Yeah. Yes. So these are the type of measurements that you get.
Yeah.
So,
yeah. Yeah. So that's you have what do we have there?
This is figure A is pictures. Okay, you can see the different of sperm.
So even this figure, what it shows you was before even coming to us to our lab, what they were able to do using normal bright field microscopy was to get a sense that there were actually more than one populations in terms of swimming patterns. And then I think we were able to just isolate that and be able to characterize it better. But I think even like that panel A comes from a bright field microscope.
Yeah.
But yeah, otherwise, I don't have much more to say on this paper. I think we summarized it. To me, it was really personally an exercise of how to wield a microscope properly. For my collaborator, it was a starting point to a lot of things.
Yeah, and I can see like I think figure G is interesting because you can see like the turf in illumination distance.
Yeah.
So I see. So so basically, your work was to capture for this this is how you capture basically the ones that are very, very close to the glass with that turf illumination surface.
And once they get sequestered in that space, that's when they start exhibiting a different type of motion. And I think that was very surprising for the community, for those who are interested in this. And I kept on looking at this field a little bit, and it's very interesting that you increase the temporal resolution of these measurements. I think when I was doing this work with Reza, we were capturing images every 100 milliseconds so that we could have enough signal. But now imagine if you're like capturing it in 1 millisecond, 10 millisecond, I don't know. Well, you might be able to see more. You might realize that if in that slither swimming, you may have averaged a few more things. Maybe it's slither, but that there was a slight tail motion. So I think temporal resolution might give you more. And if I remember correctly, there was a group that had shown that there might be a bit more to the slither.
Yeah.
But it was correct enough at the 100 millisecond temporal resolution.
Yeah, which is pretty high resolution. 100 millisecond is not nothing. But you might be missing, as you said, like some transition states between different types of swimming. Yeah, that's super interesting. I didn't know that. I found it like super fascinating that sperm can change the way they swim depending on the viscosity, for example. That probably has some very important implications for fertilization.
Yeah. And I think the group, David Sinton's group in Toronto as well, is working on that, and Reza is working on that independently. I think the key is going to be now, how do you actually do that selection? Now you're starting to have the metrics. Now they need to figure out the ways of isolating them, perhaps through the astute use of microfluidics. I'm not sure.
Okay. Beautiful demonstration. Yes, it's a beautiful demonstration of the importance of methods.
Yeah. It does not give you the solution on how you're going to be able to leverage that in the real world yet.
Yes, but it gives you the ability to measure what you're interested in, and discover new things. That's great. Do you want to move to another paper?
Sure.
Which one do you prefer?
Let's go to 2019 if you want. Let's just do it in chronological order.
Chronological order. That's the scientific reports?
Yeah.
Okay.
And I think it's going to be a very similar flavor to what we just discussed. All right. So this one. So as I remember, I said a lot of the biology came from collaborations in our lab. This is another case, but this one, I'm a co-first author with Sina over here. And so this is a lab that in an institute that does heart research.
Yes.
And
just for the people listening, I'm just going to the title is Nanoscale Reorganization of Sarcoplasmic Reticulum in Pressure Overloaded Cardiac Hypertrophy visualized by D-Storm.
Yeah.
Published in 2019 in the Scientific Reports.
What does that title tell you? What do you get out of it?
I get out of it that, as I said, I'm not a microscopy guy, but it's what I get out of it is when I see nanoscale organization of sarcoplasmic reticulum in pressure overloaded cardiac hypertrophy visualized by D-Storm, that title tells me that you're going to measure probably proteins in the sarcoplasmic reticulum or around the sarcoplasmic reticulum, and how they behave when you get muscle hypertrophy inside the heart.
Yeah, that's exactly it. So I think this is a group that day in, day out, what they look at is disease phenotypes.
Mm-hmm.
And I think one thing that they're interested in is cardiac hypertrophy. So when your heart enlarges as a function of chronic disease, where the heart eventually loses the efficiency it once had in pumping blood, where perhaps at the sarcomere, which is that fundamental unit of contraction, the molecular dance that needs to be happening is perhaps not happening the way it once was happening. So this group came to us and said, well, you're able to do super resolution microscopy. And in this case, the D-Storm stands for direct stochastic optical reconstruction microscopy, which is in the same family as the one I talked about earlier, photoactivation localization microscopy, the one where we're using 405 excitation and then bleaching out the photoactivatable FP, say with like a 500 excitation, 520 excitation, whatever it may be, or 560. In this case, you're using organic fluorophores. So the usual suspect was this Alexa fluorophore 647 that was used in most of those early papers. And what you're doing in this case is that you're just exciting your sample with a very, very high laser power, matching the excitation emission profile of your fluorophore to get it to blink. And you're just using these photo switching buffers as a way to get your fluorophore to be mostly in an off state, what we were talking about earlier. So it's the same principle. You want to isolate the emitters so that you can reconstruct an image with a localization precision that is maybe 10, 20 nanometers, so that you can get a resolution that is 20 to 40 nanometers in X and Y. And maybe something that they did not really talk about earlier is all these methods are great in 2D in X and Y, but the Z dimension is something that requires a few more optical tricks. You can use lenses like a cylindrical lens as a way to misshape intentionally your point spread function, as a way to try to capture where is that spot in 3D. But you do so at the expense of losing photons, which then impacts your localization precision. And the resolution you end up achieving in Z, it is just worse than XY. So in many ways, sometimes you just decide, well, I'd rather just, depending on the question, of course, if it's one where you need the Z information as a way to capture the ultrastructure of something, you might be stuck. But in the exercise of asking yourself, like we did here, is a disease phenotype impacting the spatial distribution of something meaningfully in 2D, then you can address it. You can address it that way.
I see.
So go ahead. Yeah.
So basically what you're saying is you can do X and Y fairly out of the box. However, if you need Z, you're getting your Z, but you're decreasing the strength of the signal by doing that.
Yeah. And there's just no free lunch in this equation.
Okay. So how do you manage that? Do you just increase you get a more powerful laser?
That's one way. That's one way to do it. There are other methods, I think, that are more elegant than adding a cylindrical lens. there are microscopy groups that are even more on the bleeding edge, like Eric Betzig's group, that are doing all sorts of volumetric imaging and have really gotten to the point where they barely have to compromise photons to be able to get to get that really good Z resolution. So it's not unsurmountable, but it requires party tricks that are non-trivial.
Okay. Nice. So which figure should we move to?
I think for the sake of just demonstrating what we did, we can just do 1, 2, 3.
Okay.
I think this one is just an introductory figure where we're looking at these 4 key proteins. There's DHPR, RYR, PLN, and CERCA, and they're just all related to having these cardiomyocytes be able to contract as they should. And I think the first one is just the wow factor, which we could do back in the day when everyone was super resolving everything that they had available. It's like, well, if you looked at this under normal diffraction limited microscopy, this is what you're getting at the top, the blurry images.
Yeah, this one.
Yeah, or if you go underneath, just the actual real images. Those are just,
okay.
Yeah. And underneath, you have the super resolved image of the exact same cell that shows you that nice striation that has to do with the contractile unit of cardiomyocytes. So that was just the first thing that we showed here, is that we're able to really, really define nicely the sarcomeric structure of those cells.
Yes. Okay. Really nice.
So that was just a panel for the wow factor. And then eventually, you still have to do some quantification.
Yes.
So the quantification, basically, if you zoom in, if you go to the other figure, sorry, you can even skip 2. You can probably go to 3 directly, because I think that will just show everything that we need to show.
This one?
Yeah. So we were provided with, so the collaborator provided us with cells that were non-diseased and cells that had come from animals that had that were models for hypertrophy, and that was, I think, achieved by intentionally constricting a very key blood vessel. I think the key aorta as a way to enlarge that heart. And eventually there was a lot of sample prep, very non-trivial sample prep that was done by our collaborators as a way to get us these cardiomyocytes in a very good state. Some immunofluorescence to the specific proteins, making sure that we have antibodies that are specific, And here, so you have the sham, which is supposed to be control. You have 2-week TAC. I think transverse aortic constriction is TAC. And then you have 4 weeks. And the more diseased one is the one on the right. And really what you're seeing, and then if you can see that's even more valuable without me saying anything, is that you're going from a nicely striated motif to something that increasingly gets disordered, where you can see that the lines are getting a bit all over the place, that your proteins are moving to the spaces in between. So really what the improvement in spatial resolution afforded us here was, look, that phenotype that you're seeing, that disease is actually impacting the spatial distribution of these key players in enabling contraction. And there's disorder now taking place, and they're not neatly organized as a way to achieve their function. And that's what we're showing here. And the way we quantify this in this paper is that we use this clustering method called Voronoi tessellation, which is basically a way of doing, it's an old mathematical method, first of all. So we're applying something really new to something really old to something really new. But it's a way of dividing points where you're tracing a bisector to each and every nearest neighbors. And you're repeating the process, and eventually each point becomes enclosed within a polygonal region, and what you're getting is a distribution of median distances for each point relative to all its neighbors, And that allows you, from that distribution, it allows you to get a sense of how your points are distributed relative to one another. It's a good method because you don't have to necessarily make any assumption in regards to how it works, but like other clustering methods like replay K clustering and so on and so on. So this one is not biased at the beginning. You're making a decision based on your distribution.
Mm-hmm.
And so then what we did was we if you go down, Tarek, maybe there's I think a figure that shows this clustering I'm talking about. Sorry for just telling you those figures, and then here. Yeah, so you can see that you segment out the cell, and then in green you're capturing now these clusters using the clustering algorithm that I was talking about. And what you can do is that you can start looking at their size. Are they more merged? Are they more isolated? So that was one thing that we did. So as you disrupt the phenotype, how are they organized along those lines? Mm-hmm. So you just report on that, first thing. And the second metric, if you go down into the last figure, is I think that one actually. We do go up.
Figure.
Yeah, 8. This one.
Yes.
Yeah. So this one we asked. Cool. We're able to measure how we're modulating the sizes of the clusters. Mm-hmm. But to what degree are we impacting the actual distribution in 2D? So we just use a 2D Fourier transform to see to what degree that periodic pattern that you would expect has been disrupted. So in this paper, basically, it was just an exercise of resolve the stuff, report on cluster on changes to cluster characteristics, and use a 2D Fourier transform on which you may have a better technical grasp than me on the fundamentals, to just tell you to what degree you've now impacted how those spots, how the proteins are organized relative to each other. So we do these two metrics. Then you ask yourself, okay, does that relate to some degree to why the cells are doing their job less efficiently? Mm-hmm. And this was good work. And I had I stayed longer, I think there were so many routes of investigation. You could have after this, you could have asked, okay, well now, have I disrupted the self-association of one player to another? Have I prevented the heterodimerization of one player to another? I think there were a lot of things that we could have further deconvolved, and this was a starting point. I believe there was a follow-up paper to this by like a few years later by people in the lab, but a lot of relatively low-hanging fruits remaining in this field by using this method on questions related to disease phenotypes in heart research.
Are these methods expensive?
Which methods?
These ones, like G-Storm.
Well, the cameras are expensive. Mm-hmm. They become a bit cheaper since, but the cameras we're talking about at the time, at least for the EMCCD cameras, I don't remember the exact number, but probably like $80,000 per camera. That is already prohibitive.
Okay. Yeah. How many cameras do you need?
For this work, just one.
Okay.
For other work that we're going to talk about too. And if you want to have really good lasers, and by that I mean lasers that are going to behave, that are going to be lasing properly, that are going to be stable, that are going to be exciting within a narrow range and not something all over the place, you also are probably going to spend more money. So if you want a high-end system, it's going to be expensive. If you want a system, it doesn't necessarily need to be. Okay. Now, the related analysis methods, all of this is relatively computationally cheap. You can run that on any laptop today. Although at the time I had to go to the analysis computer in the lab to run it, but that was 2014. I think a good MacBook Pro can do a lot of this work.
Yes. Yeah, probably. It was that was 10 years ago.
Yeah.
Computers, have come a long way since.
Yeah.
And when it comes to the antibodies for immunofluorescence, are there specific antibodies that are more expensive than other types of antibodies?
Yeah, I mean, as you know, you have polyclonal or monoclonal antibodies.
Yes.
And I think that you want to, in any case, you want to validate internally your antibody.
Mm-hmm.
And you do that through knockdowns or knockout studies as a way to see if you still have the signal relative to your protein of interest. Mm-hmm. And I'm sure you're aware of all the reproducibility crisis related to using antibodies for research. Mm-hmm. But the team here literally couldn't have done more QC to make sure that we had, at the time at least, the best available antibodies to label these 4 key proteins. And I remember, now that you're asking me, some of the decisions we made in regards to which protein to go for were tightly related to the availability of antibody. So it's not as if we decided we're going to pick these 4 guys and we're going to stick to them, we're going to make it work at any cost. It's which ones would realistically allow us to probe it, knowing that we have high, or as high as you can get, of a specificity of labeling.
I see. We should probably introduce what antibodies are.
Sure. Do you want to do that?
I could do that.
You're the immunologist here, huh?
They come from immunology, but we're not using them in that way in this case. It's like we're using them for imaging. So you're the specialist.
Well, in my case, you have a heavy chain, a light chain, you have a variable domain, and you don't need to necessarily use antibodies. You can use Fab fragments, which is a smaller part. You can use also nanobodies, which are the antibodies that are found in llamas, alpacas, sharks.
Sharks, yeah.
But antibodies. So now you can talk to us about epitopes if you want. I gave you the...
Okay. We could do that. So, but it's conjugated antibodies, right? So,
well, in this case, the primary antibody is not conjugated. We then add a secondary antibody to amplify the signal.
I see.
So that you have enough to resolve.
Yes. So you have one antibody that basically goes and fixes itself on your protein of interest. Yeah. And then you have another antibody that fixes itself on that antibody.
Exactly.
And that antibody is fluorescent, like the last one.
Yeah. And you have multiple, actually, that bind on the primary antibody.
Yes. And do they have different way? As a way to... Yeah, sorry?
No, go ahead.
Do they have different wavelength? How do you compensate for the different wavelengths? How do you...
In this case, we did this work sequentially. So we used one fluorophore, this Alexa 647 I was talking about. Mm-hmm. So we only ever had one fluorophore per sample. Mm-hmm. So we would image Circa, then we'd image PLN. So different dishes, different actual physical samples, There's a world where we could have done 3-color imaging, 4-color imaging, but the thing that we wanted to be very careful about is, given the fact that each fluorophore is going to give you different photophysical characteristics and slightly different resolution, you're going to be biasing your study against that. So you want to keep that fluorophore the exact same, so that the results are comparable between conditions.
I see. Yes. Just to give a little bit more context, if you have an antibody that's a fluorescent antibody, the light emitted by that antibody is not like it's not a line, it's not a spike, it's a wavelength, like it's a distribution of wavelength. So if you have 2 different antibodies, there could be an overlap in the wavelength, and that obviously complicates everything. This is what we refer to as compensation. So it's way easier to do the antibodies sequentially than to do them all at the same time.
But the point I was alluding to is that even if you're to have the very good spectral discrimination, separation, even then, the intrinsic properties of the chromophore that you're working with are going to yield different number of photons, which is going to yield a different localization precision, which is going to yield a different resolution, which is then going to impact the cluster analysis and then the other downstream analyses. But you're completely right. When it comes to being able to couple together a few conditions, at most, if you want to do it cleanly, it's like 3-4 colors. There are commercial instruments that allow you now to do presumably 10 colors. But then when you really dive into it, it's like, well, 10 colors, if you use this one bandpass filter and you don't use that fluorophore, but you use that one, it gets very specific. There are methods like hyperspectral filtering that allow you to use one excitation and get 2 emissions out of it.
Okay.
So there are ways. It's like you'd be able to have something in the 647 emission, something else in the 670. But again, those methods rely on things like I'm imaging something in the mitochondria, something else in the nucleus. I know that they're spatially distinct. So you're already deconvoluting them by knowing where they should be.
Yes. Yeah. And that's also a good example, the antibodies. It's also a good example of something in biology that serves a very definite purpose, but that has been completely repurposed for doing something else in research. Yeah. So an antibody basically is just, I'll simplify here, but it's a protein that fixes itself on something. And the thing it fixes itself on basically depends on the antibody. So if you can design an antibody that fixes itself on the protein of interest, you've done half the job. The second half is just to have that antibody or something conjugated to that antibody that can emit the light so you can measure it. Yeah, very nice. Anything you'd like to add about this paper, or should we move to the next one?
No, I think we said everything. I think it was again an exercise of robustness, to be honest with you, from everyone, because the methods of isolating the cells, the antibody, the choice of antibody that we were just talking about, making sure that the measurement time and time again was the same, and just having an analysis method that was robust and tailored to this. It was not necessarily the most groundbreaking paper, the most groundbreaking work, but I think it was a tight operation that we ran.
Yeah, and that ties really well what we were saying at the beginning, is like the importance of the technical knowledge. For science, because
and long hours, because the time the collaborators would, I kid you not. Like on Friday, drop me, I don't know, tons of samples, and you have to go through them before they degrade in some way. And there's no other way than brute forcing this exercise. So I would have Fridays and Saturdays, 8 a.m. until 10 p.m. just to make sure that this is well captured. So the part of this equation as well is the brute force.
Yeah. Being able to spend long hours in the lab is absolutely necessary. It's not something you can live without, and there's a very good reason for that, is because simply you're working with cells, and cells don't have weekends. they just do whatever cells do, and if you leave them too much time on the bench, or too much time in the incubator or on dry ice, they become different cells. So you have to do the work when you need to do the work, and it could be anytime.
And just for context, these were fixed cells. They're fixed in 4% PFA. But despite that, when you work with these kind of samples, imaging at T0 or T plus 2 days, there might be a slight difference in the quality of your sample. So you just want to keep it as tight as possible.
Yes. Yeah. And this is extremely important when it comes to research. From the bioinformatics side, I can tell you that I've worked on many, many samples, and I can see clearly if the experimental work has been done properly or not. You can see it with some very specific signature, for example, heat shock proteins and things like that. I know you can see from the data if the experimentalist has done a good job or not. And and that plays a humongous role when it comes to reproducibility and the quality of the results. It's like
You're completely right.
Yeah.
And what I always tell people when they're like, how do you get such nice images? Well, it's not the microscope. It's we made sure that the sample prep was as good as it can be.
Yes. Yeah. It's rarely the instrument.
Yeah.
It's 99% of the time, it's the person that did the experiment. And you can even see, and that's funny, but you can even see the level of experience of the people from the data. You can clearly see this experiment has been done by a very experienced postdoc. This experiment has been done by a first year's PhD. It's like night and day.
but at the same time, you never know. Like sometimes you have first year PhDs that are extremely meticulous. Yes, and then you have a postdoc who somehow is a master of good enough.
That is true. Yeah. That is true. I'm simplifying, of course, but that is true. It's what you see is the level of mastery and precision and meticulousness that has been applied to that specific experiment. You can see it.
Yeah.
Yes, and it's it's also funny because you have. I saw that in some labs you have some people are so good at doing some types of experiment that they get them all the time. Like say all the time doesn't exist, but like 99% of the time. It's but when someone tries to do the same experiment, they get different results.
Yeah. And I think going back to methods development, to me, a method has reached maturity once you can pass it on to someone else who's never even dabbled in it, and it works.
Yeah.
Otherwise, it remains a Cirque du Soleil performance. It remains, you have these very highly trained athletes who are doing this in a very specific milieu, with very specific lighting, and that's the only way it works. So then at that point, I'm like, it's a method, but it's a method that works in that space, that day, you know?
Yes.
So while I think there is importance in high quality craftsmanship, to democratize it is quite an exercise.
Exactly. And that goes back to the training. People need to be able to spend enough time in the lab to be just to be trained enough, because the thing is a method is so as we basically said, the method is basically as good as the people doing it. Yeah. And but the thing is, there are things. Not everything is written. It's not like a program. Like if your method is a computational method, you can write basically the method and people can run it. But if your method is experimental, there are some things that you absolutely need to know, and that are they're not always written in the methods. And this is a very important part of mentorship. You need to be able to spend enough time with people in the lab that can teach you these things. Yeah.
There's always a trick hiding somewhere. There's always something to pay attention to that you wouldn't otherwise, for sure.
Yes, and as we all went through this, but when you start your career as a researcher, you do some things that you would never do, because you would never do later during your career, because you know they have an impact. So being able to, someone has to tell you this stuff, or you have to make the error once to see it. But I
mean, I'll tell you. Sorry if I'm interrupting you, but I'll tell you something very simple. Fixing a sample. And relying on PFA, paraformaldehyde from last week, or permeabilizing buffer from 2 weeks ago, and wondering why things are not working. No, just make your buffers fresh and remove every piece of depth.
Yeah. Same thing for the antibodies. Just use fresh antibodies. It's like, well, my staining doesn't work. Maybe you use an antibody that doesn't work anymore. So that's part of it. Another thing that can be, that's also, it's just to give people an idea, is the season can play a role.
Yeah.
because because the sun is not at the same place all the time. So maybe and someone made if you do the experiment, for example, in January, and you try to redo the experiment in let's say 2 months later because you want to reproduce it. Maybe when you do it 2 months later, at the exact specific time, the sun will be hitting you a specific like this where your cells are on the bench, and that will have definitely an effect on your results. So you have to be careful of these type of things.
And I will add even to it. So the sun is shining in a specific way. It's summer in Toronto. I'm happier because it's sunnier and because I'm playing more tennis. Is that impacting the quality of my experiment? Probably.
Probably. Yes, probably. Probably. And this type of stuff, you cannot write them in the method.
that would be quite a thing if we did, huh?
Yeah, that would be. There would be like pretty long methods, and they would be also very specific to people. It's like you have to do this after lunch, or don't do it after lunch, because when you do it after lunch, you're more sleepy, so you tend to forget. So, but this is all part of mentorship, basically. Like you need people to mentor you to these different aspects of the technicalities of doing research.
But one thing I'll add, Tyler, if you don't mind, is just
yes.
There's all of this, everything we just described. But I think you have to have a plan, and I think that's what I did learn, at least in grad school. Things are going to fail. Things are going to be weird. You won't know which one is the reproducible result. So eventually you have to show up prepared, and you have to show up with, okay, I have to run these 2 experiments. I have to have 3 biological replicates. What does that take? All right. So now you're planning 50 experiments. You're planning, you just have to have that line of sight with that bonus experiment that's also going to take into consideration that other thing. So that eventually you're compacting this in time, and you're iterating as fast as you can to eventually get you to the point where you know exactly what you're doing, and you can just turn it into a routine reproduction of results. That I would say, I agree with everything we said, but the planning part can make it just go so much faster. You can parallelize so much. You can just getting in a place where you remove all the black magic out of it.
Yes. Yeah. Very important point. Very important point. And a point that's usually not discussed enough, in my opinion, is like, have a good plan. It's before you start doing anything. Like even I think this is like probably the most important thing in research is knowing that the plan can change. Obviously, when you're doing an experiment, the plan shouldn't change very much. But just what I'm saying is always have a plan. Could be for an experiment, or could be for writing a paper. Don't start writing a paper without having a plan for the paper. And the same thing is like because research is, if it's not like it's very different from school. It's you are researching things, and you're trying to find things that nobody knows. Which means it's an exercise of dealing with chaos. So if you need to be able to bring as much order as you need to be able to deal with that chaos, because if you're working in a chaotic way, there's very little chance that you will be able to discover this very hard reproducible results that you can infinitely put in a paper. It's
completely.
So the plan is
what is the expression? Luck rewards the prepared, or something like that.
You make your own luck. make your own luck, and having a good plan is a big part of making your own luck in the research.
And I think that discipline. It's like I'm going to use a football analogy. I promise you I would. It's like having a good striker. A good striker is someone who's like so disciplined in their movement, in their scanning of the game, that eventually the reason why they were able to stick their foot to put the ball in had nothing to do with luck. Is because of the positional awareness, the fitness, just being, optimizing every variable, so that eventually when the ball bounces haphazardly into the 18-yard box,
Yes.
There's someone in there.
Exactly. It's basically, it's instead of having one chance in 10 million of striking, you get 99 chance over 100.
Yeah.
That makes a big difference.
Yeah.
Yeah. Especially that you, as you said, if you play football, and we're talking about football the football that you play with your feet.
Yeah, the real one.
If you're a striker, you need to be a reproducible striker. It doesn't matter if you got lucky one day and then scored an amazing goal because I don't know, because the wind helped you. Was flying in the right direction that day. You need to be able to strike reproducibly, otherwise you're not a good striker. And the only way to get to that level is practice, reproducibility, and basically have a plan for training. It's like years and years and years of training that brought you to that level. Same thing in research, because you have to count. You don't start counting when you start your PhD. Obviously, PhD is the most important part of your research career, but it started way before that. It's actually years of training and practice and all that. But the planning aspect, I think, is usually very, very hard for people to by themselves come up with a good plan when they start their research career as a PhD.
It's tough. It's really tough.
Yeah.
I was personally, like, one thing I think that we did not necessarily highlight in this conversation, your upbringing, your parents plays a massive role in how you're going to approach problem solving later. Yes. When I think about my childhood, my dad was an agronomic engineer and an economist. My mom, a lab manager and a botanist. And already you're like in an environment where it is normal to think and to troubleshoot and to ask questions and to wonder why. And, I think I never forget that the reason why I was able to do everything is because of that in many ways. Because it's almost, at that point, it's almost not innate, but it's just deeply embedded in you. And then you're using your PhD experience as an amplifier. So I think most people don't necessarily have that base or that opportunity. And personally, I try to do good work out of respect for the fact that there is unequal opportunity given in this world and on this earth. But what I was trying to say is, even before the PhD, you have to have educational systems that teach critical analysis. Just being able to face ambiguity, being able to deal with that. And I don't have a solution for it. It's way beyond my expertise. But I think it starts there.
Yes, definitely. And it's something that's completely orthogonal to getting good grades at school. It's something different.
Yes.
It's another set of skills. It's another mindset. And it also, I think what you're saying here is very true, and it highlights the importance of mentorship during the PhD, because your mentor is the job of the mentor is also to give you that structure, that way of planning, like teaching you how you can get these plans, so you can basically you too become a researcher, and then one day, if you want to, also mentor other researchers. But it's because, as you said, not everybody has the same opportunities.
No.
A lot of people who come and start PhDs, they basically got accepted because they got good grades.
Yeah.
And that's not the same set of skills.
and this, the socioeconomics determining a lot as well. I mean, having done my PhD in Canada, having done my postdoc in the US, when you look at the crowd you're surrounded by, it's a lot of them have parents who are professors. A lot of them come from socioeconomic backgrounds that have allowed them to explore.
Yes.
The amount of scientists that are not becoming scientists just because of socioeconomics is ridiculous.
But I would say that this is definitely true in North America. I mean, from my experience at least. But in Morocco, it's different. And this is one of the aspects I like about EM6P is that we get PhD students from basically everywhere in the country.
That's great.
Different social backgrounds, and that's I think it's also surprising but because I was used to what you said. It's like the thing we have in Morocco, for example, is doctors, like clinicians, tend to become doctors too. is That's something that we have a lot. But when it comes to research, they come from everywhere, and
That's good.
and what I've noticed is the quality of the students is pretty high from all social backgrounds. But also, what's very interesting is that it doesn't necessarily depend on the grades, like how good they were, how good their grades were for during their master's degrees or their bachelors. It's a completely different set of skills. It's basically the defining factor is not intelligence, because or things like, because everybody's intelligent. I mean,
Yep.
it's like, I mean, everybody has the mental capacity of doing that type of work. It's more how good are you at dealing with uncertainty?
Exactly.
Yeah, that's the defining factor. And I also strongly believe that when it comes to intelligence is something you can train, just like any other, just like a muscle. You can become smarter if you want to. Just have work your brain. That's it. Everybody can do that. But dealing with uncertainty is a different set of skills, because it's more emotional.
Yep.
And also something you can train. But most people it's not that most people don't do it. It's like the schooling system doesn't prepare you for that.
I agree with you. I mean, I'll even use my own example. When I was in high school, I didn't have the best grades. You know, I was not a 20 out of 20 generating machine. That was not my goal.
Mm-hmm.
Of course, but at the end, for the baccalaureate, I got a bit more serious. Mm-hmm. But my point is, at some point, I think you have to be self-motivated enough to deal with the complexity and the uncertainty of it all. And that happened to me when I moved to England, actually, during my undergrad. For some reason, I got extremely serious about the whole thing. Very obsessed with what I was doing. And that was a declic in many ways, because it put me in a position where, okay, now I'm on top of it and I'm learning really fast. I'm learning how to learn even faster. Mm-hmm. But if you look at me when I was 15, there was no way to conceive that I would be doing what I'm doing now. Mm-hmm. Like most of the school advisors were probably thinking I was just gonna do, I don't know, who knows what.
Yes, I know that experience. I also know people like that. And that goes back to what we were saying, like your ability to be good at research has nothing to do with your ability at getting good grades.
Yeah.
And I think this has like important implications, and we should, in my opinion, we should rethink how we train people before the PhD level, getting them these critical skills that will allow them to become researchers if they want to.
Yeah. I agree.
Yes, and also these skills are pretty good, like for life in general. It's like being able to deal with the uncertainty and making hypotheses and then moving along with the chaos and still finding something valuable. I think it's a very valuable set of skills for life in general.
It's the story of life, yeah. You have to be ready not to be ready.
You have to be ready. You have to be ready regardless of your level of readiness. You're always ready. Yes. Do you want to take a break?
Up to you.
It's up to you. We could take a break, or we can just move on with the next paper.
I'm ready.
Okay, let's move. Let's go. Let's do the next paper. So which one? The JCB or the nature methods?
We can go briefly over the JCB, but I think I would like to spend more time on the nature methods one.
Yeah.
Since it's the most recent one. And I think the JCB is like more of the same. So I think the nature methods one is going to be cool.
Mm-hmm. Perfect. So give me 1 second. I'll open the JCB. Can you see it?
Yep.
Perfect.
So I think for this one, all you need really is that first figure, which is a schematic figure.
Yeah.
And then the second one.
Perfect.
And maybe the last one at the end if we care to go to those details. But if you go to figure, so let's start with the title. You like to do that, sorry.
Yeah. So, because for the people listening, the title is Correlated Storm Homo-Threat Imaging Reveals Highly Heterogeneous Membrane Receptor Structures. And it was published in 2021. Amine is the first author, so it's Driouchi et al., and it's in JCB.
Yeah, so that one takes all the parts for everything we've described before. It takes the D-storm we just talked about. It takes the turf that we did for the sperm study.
Okay.
And it adds this layer of this method called Forster resonance energy transfer, which is basically when you have fluorophores that are separated by a 1 to 10 nanometer distance, there is some amount of energy transfer that gets, in the case of two non-identical fluorophores, the excitation emission to shift. So there is an absorption in energy that is getting the spectral properties to be modulated. In this case, this is not what we're doing. Okay. In this case, we're doing homo-threat, which is two identical fluorophores that you're exciting using polarized light. With light being a wave, you use a polarizer to polarize it in one way. And what you're doing is that you're relying on the fact that here we're interested in self-association. So a protein being a monomer, forming a dimer, a trimer, and oligomer. And what you're saying is if the fluorophores are in very close proximity, there will be an energy transfer that is going to lead to the excitation light to be depolarized, to be polarized in another direction. Whereas guys that are standing on their own, that polarization is going to be kept. And don't ask me the details, I don't remember them in detail myself. But the idea here is you're capturing that polarized emission and depolarized emission in two distinct cameras. And then you're calculating a pixel-wise ratio that tells you about the enrichment in depolarization and polarization, which is indicative of the enrichment in monomers and oligomers for this protein of interest that we're looking at. The biological problem that we were interested in, and this is like a 4-year-long project, by the way. So Scott Gray-Owen, who's a professor in molecular genetics, was interested in this protein called CCAM1, so carcinoembryonic cell antigen-related cell, what is it called? Cell adhesion molecule, I believe. I may have butchered that a bit. It's a transmembrane protein that is linked to infection, differentiation, development, is overexpressed in certain cancers, is downregulated in other cancers. So there are a lot of questions in regards to it. And it exists in multiple splice isoforms. And the one that we are looking at here is the prototypical one, which is the CCAM1 4L. 4 standing for the number of domains in the extracellular domain. Then you have a transmembrane domain, and the L stands for the long cytoplasmic domain, which I believe is a 76 amino acid chain. The question we asked was, can we build a method that tells us within a cell how do they spatially distribute? So the only thing that we knew before starting this investigation was that the CCAM proteins clustered. They formed clusters, and then there are some regions that are more diffuse. Yes. Then the question became, what are those clusters made of? What are the diffuse regions made of? And can we resolve that? So with D-storm, what you can see there at the bottom, you're getting the spatial localization with that resolution. That is, let's call it 30, 40 nanometers. With FRET, you're getting a pixel that gives you a value. So yeah, that's the storm. And the FRET, you're getting a pixel that gives you a value where, say, the red pixel of 0.5 tells you, in that pixel, I have an enrichment in monomers, polarized emission. In the blue one, no, it's depolarized. And then the idea was coupling the two methods in the same measurement, the same sample, to be able to correlate the two methods and say, well, it appears that regions that are more clustered are actually more monomeric, while regions that are more diffuse are more oligomeric.
Mm-hmm.
So what this required to do, if you go to the next figure.
Yeah. Before you move to the next figure, I think I made a mistake. Yep. It was published in 2022. It was received in 2021, but it was received in 2022. And I believe I said JCB, it's JBC.
I forget myself, you know.
It's Journal it's Journal of Biological Chemistry. Yeah, Biological Chemistry.
And this is just the transmembrane protein I was talking about, and it's forming dimers and it's monomers. It's forming also intercellular interactions, and that was just to show that calmodulin is able to break the dimers. So there are some cofactors that are important. But if you go to the next one, yeah. So that's just the FRET that I was talking about. And, you know, if you look at CDE, you're able to capture the polarized, depolarized emission. And then the trick here, we're talking about the troubleshooting. You have two cameras that are receiving this emission. You can imagine that there will be a problem there. It's the camera alignment, so that you can calculate these ratios. And we had them in a position that's orthogonal. And you have this cube that is sitting in between the two cameras that is splitting the emission. So you basically have to have a design, a setup that allows those two cameras to always be kind of very well aligned. And the reason I'm saying kind of and not very well aligned is that you still have also to do some registration, some image analysis to register the two images to one another to make sure that the pixels that you're for which you're calculating a ratio are the exact same ones. And it's quite a contortionist effort to make sure that, again, the day in, day out, the cameras are aligned, and you can do that with, like, you take beads, for example. You can, there are easy ways to make sure that your cameras are aligned. You also want to make sure that your cameras are as sensitive as each other, that there isn't one where the chip on the camera has lost sensitivity and whatnot. You want to make sure that they are at the same level of calibration.
Okay. And how do you use beads for that?
Yeah, so you just make sure that, you know, you have some distribution of, or something structured, like let's say beads. You're imaging them in both cameras, and you're just moving one camera relative to another to overlap the two images. Yeah, and you can just go ahead.
No, it's just like you have two images, and then you move the two images until they overlap, basically. So you have like dots, like for example for the beads, and then the beads need to overlap. And this is how you calibrate before you start working. Yeah,
and you can imagine that's crude. That's still quite crude. And then eventually, once I get to my exact cell, you may want to do a small nano adjustment, but there's only so much you can do with you know the adjustment knobs that you're provided with.
Of course, because this is like we were nanoscale.
we're calibrating for nanoscale with our pudgy fingers. Yeah.
Yeah, because you just like have a dial. And you can, good work. Okay, so you have these two cameras. Yes, please go ahead.
Yeah, no, and then if you go forward, I think one thing that's going to be interesting here, if you go to the next figure, is how do you do this exercise of correlation? So, you know, you and I were talking about antibodies earlier. So,
we have primary monoclonal and secondary polyclonal.
That's right.
Yeah.
So what we did here, instead of doing that classic add a primary and add multiple secondaries with multiple fluorophores, our FRET-based method depended on this enhanced yellow fluorescent protein. There was previous work that was done in the lab where we knew that it worked well with that fluorescent protein. And we had already the plasmids and the constructs ready. So I didn't do that. But what we had to do is figure out a labeling strategy that would allow us to correlate the storm and the FRET imaging. And at that time, nanobodies that we talked about earlier briefly became available. And then you could have a nanobody that could be anti-XFP, whatever FP you're looking at. So then, spatially, instead of having a cloud of Alexa 647 sitting on top of the secondaries, just like you have on the left there, you have one fluorophore 1 to 2, if I remember correctly, sitting on one nanobody in close proximity to the yellow fluorescent protein. And that was very important for us to be able to correlate the two.
I see.
And then the rest of the paper is really more of what I've shown you before. So having done that, you can go into the last figure. Yeah, you can skip this. It's just technical stuff about fixation and permeabilization. Perhaps figure 5, I believe. Yeah, this one.
7, yeah.
Yeah. 7. So this figure, I think, summarizes the whole point of this exercise. So panel A, you have a cluster of CEACAM proteins. We knew that. B, you now have a pixel-wise anisotropy map, this concept, this enrichment in monomers and dimers. And so it shows you in red the regions that are more monomeric, in blue, green, the ones that are more oligomeric. And then in C, you have the super resolved image, the storm image. And in D, you have again that cluster analysis method that I became very fond of, the forward node tessellation. From that, you can extract sub-features like smaller, dense clusters. So you have bigger clusters in blue, and you have smaller ones in green that we call nanoclusters in this paper. And then if you go down to the bottom part of this figure, you'll see that, for example, this region that has this distribution, actually, which is pre-oligomeric or pre-dimeric, has this set of nanoclusters and whatnot. But basically, in this paper, and I think we have that in supplementary figure where we compare different regions, the very counterintuitive finding was that regions that are dense are enriched in monomers, and regions that are diffuse are enriched in oligomers. You would think that makes no sense because, they're getting close to each other. They're probably more oligomeric, but we ran every control and made sure that was not an artifact of something. And we reported on its results, and we're pretty confident that what we're measuring here is these CEACAM proteins do indeed form monomeric clusters and have diffuse dimers, oligomers that dissociate to go into these monomeric clusters. And we had, it's not in this paper, it's in my thesis, like point mutants to further validate this.
Maybe we should give a definition of what a monomer and oligomer is.
Yeah, so when you have protein folding, it's a protein folds from a primary sequence to a set of secondary structures that has alpha helices and beta sheets and loops, and that eventually all comes together to form a folded protein, a tertiary structure. And that tertiary structure is at times not the functional protein. Like in the case, there are many proteins that end up associating with themselves as a way to give rise to their function. Some of them will need to form a dimer, so 2 of them, a trimer, a hexamer, as a way to give rise to their function.
Yes. So basically, just to summarize, you have the tertiary structure is basically the 3D of the protein.
Yeah.
The 3D structure, there's basically 4 levels of structure. Like the first one, just like the sequence. The second one is basically closeness of the different parts of the protein. The third one is the 3D. And then the fourth one is when you take these 3D proteins and you put them together.
Yeah.
And like a monomer is like a protein by itself.
Yeah.
And if you have more than that, then this is basically like either the protein can basically form complexes with other proteins. We can also form complexes with itself, and then they start becoming active. They're like building pieces of something bigger, like a bigger machine.
Yeah.
So so just that was more to explain why it would be counterintuitive, basically, so people can understand why it would be counterintuitive. Your finding would be counterintuitive, basically.
I think if you're to just do the thought exercise of imagining a protein sequestering itself in a tight space. And if you were to just ask, in your opinion, is this now a monomer or an oligomer? By sheer physics, I think the vast majority will think that due to their proximity, they must be forming a functional oligomer.
Yeah.
And
because you're squeezing them together.
Exactly. The reality of it is they're close together, but they are not forming. They are not within that 1 to 10 nanometer distance. That would even not confirm necessarily that they have self-associated. Yeah.
Yeah. It's interesting. It's do you have an idea why?
Probably at the time. Let me try to think about it. I don't know this fundamentally why, but we do know that intercellularly, the intercellular interactions are driven by these monomeric interactions. And that you have cell projections, that those things that we call clusters are actually cell projections in 3D. And the intercellular CCAM interactions seem to be mostly monomeric. Why? I don't know. Are there some that are dimeric? We haven't necessarily measured that. As in, we have not seen evidence of that.
Okay.
And we do know that if you prevent the cells from forming monomers, you are reducing the ability of cell-cell contact. So there's a whole world there. I don't know the fundamental why, but it seems to be that the monomers are facilitating cell-cell interaction.
Okay. Interesting.
Yeah.
Yeah.
And again, it's a whole world out there. I remember presenting this at the CCAM conference in Banff, Canada in 2019. And it raised a lot of eyebrows in a good way. Like people were very curious about this. And because I didn't have the domain-specific sophistication to go into the details of the protein, I was not, the life and time of the protein. I was not able to necessarily, hypothesize and speculate on each and every dimension of it. And it's a current theme in my career. I'm never the expert in one piece of biology. Because
you make the tools, the methods, and you move from one field to another. Basically, you apply it to different fields.
Yeah.
Is this the last project of your PhD?
It was the first and the last.
It was the longest.
It was the longest.
Okay, I see. So did you finish it during your PhD, or did you have to write a paper with you for your postdoc?
I finished all the work in 2017 or something like that.
Okay.
And then I think it went dormant for some time, and we decided to finish it in 2022. That was a long time, huh?
Yeah, it's a long time for a paper.
Yeah.
So in 2022, were you still doing your PhD?
No, I was at Icon Therapeutics, which is the next paper. And it was just a matter of, the paper was already written, and it was just a matter of iterating with my advisors and, making sure that everyone is on top of it, make the final adjustments. And that, that final 1% took years.
The long tail.
Yeah. Because everyone was less vested in it.
Yes. Yeah. That's what happens. It's like when you move geographically, it becomes harder to, especially if it's a project that you started years ago, like getting everybody motivated about it. It's takes more time.
Yeah.
Yeah. But it's a cool project. It's a cool paper. So
yes, potential.
Yeah, I mean, I think it's really nice. I always like it when you have different methods put together to get a higher resolution. I like the distone plus homofret. I mean, you should probably say what fret is. We haven't said that.
Yeah, I think the fundamental principle. I don't remember fully myself, but as I said, I think I alluded to it by saying that you have this energy transfer that happens within a 110 nanometer distance and happens actually in a way where it's way stronger. It's not a linear scale. And what it does in the case of two fluorophores that are different is that energy transfer is going to change the wavelength of your emission. So you can spectrally tell to what degree those two things are in close proximity. But in the case of HomoFred, that energy transfer is happening in a manner that changes the way with which your light is being emitted. So then, as you excite your sample with a selective type of polarization, the idea here is you're selectively exciting those fluorophores that have what they call their dipole moment aligned with your with the excitation light that you're using, whereas the depolarized one is going to come out differently. Okay. And don't ask me more because I need to rebrush my fundamentals there.
Okay. So is there anything you'd like to add about this paper?
No, that's it. I think it was a good exercise again, making sure that we have a method that's somewhat reproducible.
Yeah, looks it's a very nice method.
Thank you.
Yes. So okay, let's have a quick break and then we move to the big paper.
All right.
Let's make a coffee.
All right.